So one of the things that we know that when we are explorers is we'll take plants with us when we leave the face of the earth. It's much like when we left from the East Coast for the Oregon Trail, right? We took all our plants with us, our biology, our plants, our animals, and we didn't know how things were going to be growing and living in our new environment, and so we had to take that stuff with us. So if we're going to go to Mars, or if we're going to even do long-term space exploration in orbit, we're going to need to know what it is about plants that we can help them adapt to this new and novel environment. And one of the reasons why this is important is because space flight, especially microgravity, but even on Mars, these are novel environments for plants and in fact any terrestrial organism. So we don't know how exactly they're going to respond. The way we look at their responses is through their gene expression patterns. So we look at what genes get turned on by spaceflight, what ones get turned off, and by learning that, we can be well informed about what it is that would make a plant better adjusted, better physiologically adapted to spaceflight. Plants can grow in spaceflight just fine. I mean, as far as in a microgravity environment, they do very well. But in order to do well, they have to turn on a ton of different genes. They have to turn off a ton of different genes. They still do just fine, but it's like they've put effort into adjusting themselves. Now, what if you take those genes that are causing all this extra trouble that they don't really need? It's not like they need to have all these cell wall remodeling genes. It's not like they need to have all these pathogen response genes because there's no pathogens there. It's they're turning on these genes because they don't know what to pull out of their metabolic toolbox. They don't know. It's a novel environment. So they're throwing all sorts of different things at it to say, I think this might make me feel better. But in fact, what it does is it's a metabolic cost. It costs you energy to do these things. So with all the kind of background work we and others have done with this molecular biology, what we can say now is that, all right, plants don't need these dozen genes. Let's either engineer them out or let's choose varieties that don't have these genes or don't express these genes. And so what this boils down to is the seed packets you're going to take to Mars or the seed packets you're going to grow on the space station are not the ones you pull up from your local hardware store. These are the ones that are going to have to be either engineered to be better adapted, or they're going to be part of a breeding program that also chooses things that are better adapted. So the light microscopy module, the LMM on the space station, helps us narrow down exactly where these genes are having their effects. And so we know, for instance, from our previous experiment, our APEX-3 experiment, that a number of patterns of gene expression change dramatically, especially in the plant roots, as they adapt to spaceflight environment. Now we know this on a number of different levels, but for the most part what we do is we take the plants, we bring them home, we get all ground up, and so then we do microarrays, we do RNA-seq, we do all these esoteric biochemical analyses that tell us what genes levels are expression in these different tissues. And what we found was some preliminary data suggests that in addition to the kind of usual suspects of genes that are associated with cell wall metabolism and different types of remodeling that happens in spaceflight, we also saw a number of genes that are associated with something that we call DNA methylation. And why DNA methylation is important is there are key genes that govern how plants do epigenetic adaptation as opposed to just sort of standard physiological adaptation of gene expression. And so that was one of the focuses that we wanted to have for this next APEX experiment, APEX-4. Now, part of that is using the LMM, and how we use that is we've taken some of these genes and we've engineered them so that they have fluorescent markers associated with them. So anywhere a plant is expressing some of these genes of interest, what we'll be able to see is a fluorescent spot within those cells. And so now, in addition to having the global information from the ground-up tissues that we get back, we also have specific roadmap of where these genes are being expressed and how they might impact the adaptation of plants in spaceflight. As NASA likes to say, it takes a team to launch a dream. And no truer words have ever been spoken as far as the spaceflight exploration and research goes. So we've had a lot of help along the way in order to get this kind of payload launched and ready and stuff. We're funded by NASA SLIPS. We are supported by the folks at Kennedy Space Center. And right now, what's happening is at GRC, the Glenn Research Center, this is where we come to do the final stages of the experiment, which is to log in and essentially drive the LMM, the light microscopy module, from the ground. All the astronauts have to do is they just have to put the plate in the microscope on the orbit, and then all the real work happens here. And so what my colleague Rob Furl and I do is we look over the shoulders of the GRC engineers and technicians that push all the right buttons and help us navigate around and find the valuable science that goes into answering all our questions. One of the very gratifying aspects of the kind of research that we do, in addition to the very basic research questions we're answering, is that we are fundamentally helping NASA exploration initiatives. And how we do this is being prepared to know what kind of plant biology we will take with us when we take our plants off planet, both to Mars and also for long-term exploration just in orbit.